Yeah, in today's lecture we are going to discuss about eukaryotic transcription factors. I think in the previous lecture I basically told you about the DNA binding domain. I told you that two major domains of eukaryotic transcription factors, namely the DNA binding domain and transcription activation domain. We discussed very briefly what kind of DNA binding domains are actually present in various eukaryotic transcription factors. We discussed about helix turn helix motif, zinc finger motif, leucine zipper motif, and the helix loop helix motif, to mention a few. And then we basically also discussed about the various experiments that people actually use to identify transcription factor binding sites in various promoters, and also to identify the various functional domains in the transcription factors. So today we are going to focus about the other important domain in the transcription factors, namely the transcription activation domain. Okay. So let us just recapitulate what we studied in the last class. So what basically I told you in the last class is that eukaryotic transcription factors basically contain a DNA binding domain, and it is through this DNA binding domain these factors are able to go and bind to specific sequences in the upstream region of various promoters, and this sequence specific binding of a transcription factor to the promoter is the first step in the eukaryotic gene expression in the transcription activation process. So today we are going to focus about the transcription activation domain, which is actually another important key domain in the eukaryotic transcription activators. And we also mentioned in the last class that in addition to these two domains, there are also many other important functional domains in transcription factors. For example, they have what is called as a dimerization domain. In some transcription factors, the dimerization domain may be present in the DNA binding domain, whereas in some cases it may be in the transcription activation domain. And it is this DNA binding domain which actually brings the two monomers. Many a times the transcription factors function as dimers, and through these dimerization domains the two monomers come together, and motifs like leucine zipper, helix loop helix actually try to bring these two monomers together and then project the DNA binding domain in such a way that it is now able to go and then recognize this specific response element in the DNA. And I also told you that certain transcription factors like glucocorticoid receptor and other steroid hormone receptors also have a domain for binding to certain effector molecules like small molecules. For example, in the case of steroid hormone receptors, it is the steroid hormones like glucocorticoid receptor, it is glucocorticoid hormone, estrogen receptor, it is the estrogen hormone, and so on and so forth. So you need to have some of these transcription factors have a domain for binding to the effector molecules or the small molecules, to which small molecules like steroid hormones can actually bind, and binding of the steroid hormone brings about a conformational change, and therefore now this receptor or this transcription factor can go and then carry out its functions. So since we discussed in the last class extensively about the DNA binding domain, we are going to skip that today and focus primarily on the transcription activation domain, and also a little bit about dimerization domain and effector binding domain and so on and so forth. So let us now really see what has been done and what kind of experiments were actually done to understand the functions of various transcription activation domain in eukaryotic transcription factors. Now let us first try to see how can you define a transcription activation domain. Now transcription activation domains, in this I have actually abbreviated as TADs. They are actually regions of a transcription factor which in conjunction with the DNA binding domain can activate transcription from a promoter by contacting transcriptional machinery. When I say transcription machinery, it means the general transcription factors on the RNA polymerase, either directly or through other proteins known as co-activators. So the function of a transcription activation domain is basically to contact the pre-initiation complex in the core promoter region, and this can happen either direct interactions or through certain other proteins which are called as co-activators. So they actually act as mediators between the eukaryotic transcription factors and the basic transcription machinery. Just as we have transcription activators, there are also transcription factors which, when they actually bind to certain specific sequence of promoter, bring about repression of transcription. So the regulation can be either positive or negative, and such transcription factors which actually result in the decrease in transcription activation is called as transcriptional repressors, and such transcription repressors, instead of a transcription activation domain, they actually possess what is called as a transcription repression domain. Okay. So transcription repression domains, just as the transcription activation domains, they are regions of a transcription factor which in conjunction with the DNA binding domain can repress transcription. Instead of activation, they repress transcription from a promoter, again by contacting the general transcription machinery or the RNA polymerase. Okay. So, and in this case again the interaction can be either direct or it may be mediated by molecules, in this case they are called as co-repressors. So, the transcription activation domain or the transcription repression domain either can activate or repress transcription either directly by interacting with the general transcription machinery or through molecules called co-activators or co-repressors. Now, a lot of effort has actually gone on to understand how actually these transcription activation domains or transcription repression domains actually function. Now, it is not very easy to understand is because see we know in our from our previous lectures the transcription initiation requires at least 50 odd proteins. We have RNA polymerase which itself is a multi-subunit protein, we have general transcription factors which again each one is a multi-subunit protein. So, at least about 50 to 60 proteins are actually involved in transcription initiation in the core promoter region. So, we are now talking about certain other transcription factors or transcription activators or transcription repressors which can interact with any one of these 50 odd proteins. So, if you have to understand how these transcription activators are functioning, you need to understand which component of the general transcription machinery they are interacting with. So, the studies of these kinds of mechanistic insights are not very easy and lot of effort has actually gone on to understand the mechanism by which these transcription activation domains actually function. So, what I showed in this cartoon is basically to summarize what I told you so far. You have transcription factors which contain the DNA binding domain and either a transcription activation domain in the case of transactivators or transcription repression domain in the case of transcription repressors, and when they bind to specific sequences in the promoter region of a DNA, they can either directly contact the general transcription machinery or there could be what are called as co-activators or co-repressors which again are multi-subunit complexes, and because of these interactions, when these transactivators or transrepressors interact with these mediators in turn interact with the general transcription machinery and result in enhancement of transcription of that particular gene. Okay. Now, to understand the mechanism by which these transcription activation domains function, we need to do, as I said, there are more than 2000 gene specific activators that have been discovered so far. So, what has been done is to take one or two of these transcription activators as examples and then see how the transactivation domain in these particular transactivators actually function. So, during the late 1990s or late 80s and early 90s, certain classical transcriptional activators have been chosen as model system to understand the mechanism by which they function as transcription activators, to actually study what kind of transcription activation domains these proteins actually contain. So, for example, some of the classical examples which people have been using for understanding the mechanism of transcription activation domains are in the yeast proteins like GAL4, which actually is involved in the upregulation of galactose metabolism genes, GCN4, again a very important transcription factor which is actually involved in the upregulation of genes involved in general amino acid control in the case of yeast, because as a general amino acid control means when the yeast cells are grown in a medium rich in amino acids, none of the amino acid biosynthetic genes are turned on because there is no need, because there is amino acids are present in the medium, but when the amino acids are not there in the culture medium, then all these amino acid biosynthetic genes are turned on, and therefore the organism starts making their own amino acids, and the transcription factor called GCN4 plays a very important role in this general amino acid control. Similarly, there is a protein called heme activator protein. Here, just like in the case of steroid hormone receptors, the steroid hormone acts as an activator. In the case of yeast, there is a protein called heme activator protein or HAP1 whose activity is actually modulated by heme. Heme binding to this transcription factor modulates its activity. So, some of these transcription factors were actually used as model systems to understand the mechanism by which their transactivation domains actually function. When it comes to mammalian cells, the classical examples have been steroid hormone receptors, heat shock transcription factors, nuclear factor kappa B. These are the proteins, some of these proteins we have already discussed in the previous class very briefly, and these again have been taken as examples to understand the mechanism by which some of these transcription factors are actually activating transcription. It is not only the eukaryotic transcription factors, people have also realized there are many viruses which actually infect these eukaryotic cells, many animal viruses. For example, you have herpes virus, HIV, adenovirus and so on and so forth. It turns out even these viruses actually contain transcription activator proteins or even co-activator proteins, and therefore even these viral proteins were actually used as model system to understand the mechanism by which these viral proteins are able to activate transcription of the viral genomes. And some of the very well studied viral transcription activators, for example, are herpes virus activator VP16 which is a very good model system to study some of the early work on transcription activation were done using herpes virus VP16 protein, and more recently the human immunodeficiency virus of the HIV also contains a very potent transcription factor called TAD protein, and this also has been now used as a model system to understand the mechanism by which this TAD protein actually enhances the rate of transcription of the HIV viral genome. So, a number of transcription factors of eukaryotic origin were used as model system to understand the mechanism by which the transcription activation domains actually function. Now, some of the very interesting early experiments were done when these studies were actually being pursued, and one of the most interesting experiments that were done are what are called as domain swap experiments. Now, what do we mean by this? Now, I have already mentioned to you that in many of these eukaryotic transcription factors, they contain a DNA binding domain and a transcription activation domain, and these are modular in nature. And it turns out these domains can actually function independently of each other. So, to actually demonstrate that these two domains, that is the DNA binding domain and the transcription activation domain, can actually function independently, what kind of experiments were actually done? The most popular experiments are called as domain swap experiments, wherein you take the transcription activation domain of one transcription factor and link it to the DNA binding domain of another transcription factor. That is what I have shown in this particular cartoon. For example, you have the GCN4 and GAL4, which are very potent transcriptional activators in yeast cells, and each of them have their own DNA binding domain and transcription activation domain, which I have shown in different colors. Now, what has been very interestingly done is the people have actually taken the transcription activation domain of either GCN4 or the GAL4, which are of eukaryotic origin, and they actually fused it to the DNA binding domain of a transcription factor called Lec-06, which is of prokaryotic origin. So, you have a DNA binding domain from a prokaryote and transcription activation domain from a eukaryote, and when you fuse these things and then make this kind of a chimeras where the Lec-06 DNA binding domain is fused to the GCN4 this is not DBD, this should be transcription activation domain. Similarly, Lec-06 DNA binding domain fused to GAL4 transcription activation domain. Now, these chimeric proteins, instead of activating from a GCN4 DNA recognition element or a GAL4 recognition elements, will now start activating transcription from a Lec-06 recognition sequence, clearly telling that these transcription activation domain can function irrespective of what kind of DNA domain is attached to them. If these transactivation domains are attached to a GAL4 DNA binding domain, they will activate from a GAL4 response element, promoters containing GAL4 response element. If the transactivation domain is attached to a GCN4 DNA binding domain, they can activate transcription from a GCN4 recognition element in the promoters, or if they are attached to even a prokaryotic DNA binding domain, they can actually activate transcription from the sequence recognized by the prokaryotic DNA binding domain. So, using these kinds of studies, people have actually demonstrated very clearly that eukaryotic transcription factors contain the DNA binding domain and transcription factors which are modular in structure, modular in nature, and these domains can be swapped between transcription factors and they can function independently of each other. So, using some of these domain swap experiments and many other deletion studies, cytogenetic mutagenesis and so on and so forth, people have started understanding how exactly transcription activation takes place, how exactly this TID actually function, and the outcome of all these experiments is that at least 3 major transcription activation domains were actually identified in many of the eukaryotic transcription factors, and these were classified as acidic domains, glutamine rich domains and proline rich domains. Now, what do I mean by this? What I mean by this is that the acidic transactivation domains are actually characterized by the presence of what are called as a negative charge. That means they are highly rich in acidic amino acids such as the glutamic acid and aspartic acid. So, in transcription factors like yeast, GCN4, GAL4, mammalian glucocorticoid receptor or the herpes virus VP16 transactivation domain, they contain the transcription activation domain is highly rich in acidic amino acids, and therefore these are called as acidic transcription activation domains. Okay. And very interesting experiments were actually done in the early 90s, in the late 80s or early 90s, especially from the laboratory of Mark Toshney, where he actually extensively studied some of these transcription factors which contain the acidic activation domain, and he actually created an artificial transcription activation domain which contains what is called as an amphipathic helix. This is not a natural domain, he constructed an artificial transcription activation domain, which is basically a short peptide in which the acidic residues are present on one surface of the helix and hydrophobic residues are present on the other surface of the helix. Now, when you make this kind of amphipathic helix with acidic residues on one side and hydrophobic residues on another side, and this kind of amphipathic helix or the gene coding for this peptide, when you link it to a heterologous DNA binding domain, it actually functioned as a transcriptional activation domain. So, this is actually to prove that many of the acidic amino acids present in the natural transcription activation domains, which I showed in the previous slide, like in the GCN4 Gal4, these acidic activation domains can actually be mimicked by a synthetic transcription activation domain, which actually contains acidic amino acids on one side, hydrophobic amino acids on one side, clearly indicating that presence of acidic amino acids is one of the major characteristic features of transactivation domains of eukaryotic transcription factors. So, when fused to the DNA binding domain of Gal4, these peptides, which contain an amphipathic alpha helix with acidic residues on one side and hydrophobic residues on the other side, efficiently activated transcription from chromatin containing Gal4 binding sites. These are all very interesting sites, and those times are all very exciting results, because you can actually make synthetic peptides and demonstrate they can actually function as transcription activators in a cell-free transcription system or in a transfection system. So, those are all very exciting experiments which were done in those days. If you want to really study more about this, you should actually study some of these original papers published from Tashner's group. Very interesting experiments were done. Similarly, certain transcription factors like SP1 actually contain a transcription activation domain which are rich in glutamine. Okay. So, the proportion of glutamine residue seems to be more important than the overall structure. So, just as some factors have glutamic acid or aspartic acid or acidic amino acids, there are certain transactivation domains which are highly rich in glutamine amino acid, and there are many other transcription factors like C-Jun, AP2 or OCT2 which actually contain a proline rich transcription activation domain. Now, these are just examples I gave you. As people started studying more and more transcription factor, more interesting transcription activation domains were discovered. For example, there are some transcription factors which contain isoleucine rich region. Okay. So, we will not discuss all these transactivation domains, but just remember that very interesting outcomes came from some of these mechanistic insights in transcription activation domain functions. There are domains which contain acidic residues, there are domains which contain glutamine residues, there are some which are proline rich residues. They all function as transcription activation domain when linked to a heterologous DNA binding domain, and you assay the activity of a promoter either in a cell-free system or in a transfection system in vivo. The outcome of some of the important experiments which are actually done to understand the mechanism of transcription activation, there are also some very interesting what can I say the benefits that came out of the studies, because wherever possible I also mentioned some of the benefits that came out of the basic research. For example, one important benefit that came out of this understanding that transcription factors actually have a modular structure is, we know for example from what I have told you, that when you have a DNA binding domain and when it is linked to a transcription activation domain, it can activate transcription, and if these transactivation domain and DNA binding domain are not linked, then they cannot activate transcription. They have to be physical, they have to be in physical proximity, only then transcription activation can take place. Now, using this particular people have developed what is called as a Yeast-2 hybrid assay to actually study protein-protein interactions. Now, what do you mean by that? Suppose for example, I am interested in a protein, let us say X, and I want to know, are there proteins which actually interact with this particular protein X in a particular cell. Now, this understanding these protein-protein interactions are very important, and as we study more and more about transcription regulations, you realize that the entire regulation of gene expression hinges on two things. One is DNA protein interactions and protein-protein interactions. It is these two interactions which actually govern the entire gamut of eukaryotic gene regulation. Now, so you want to now study the mechanism by which the transcription factors actually function, you need to identify what kind of proteins it actually interacting. For example, if I have to now understand what kind of protein the glucocorticoid receptor is interacting, because ultimately it has to activate transcription, I do a simple Yeast-2 hybrid assay where I take the transcription activation domain of the glucocorticoid receptor and fuse it to the DNA binding domain of a well characterized transcription factor. Okay. Then I have a yeast cell library in which they express, each cell expresses a different kind of a transcription factor, different kind of a protein which is actually linked to the transcription activation domain of these proteins are actually linked to the transcription activation domain of a well characterized transcription factor. Now, as I told you, if the DNA binding domain has to activate transcription, the transcription activation domain has to be brought in close physical proximity to the DNA binding domain. So, when you actually transfect this your protein of your interest, in this case for example a glucocorticoid receptor linked to a DNA binding domain or the transactivation domain of glucocorticoid linked to DNA binding domain. Now, and if I introduce them to yeast cells, and if there is a protein in the yeast cell which actually interacts with the glucocorticoid receptor or protein X, then because of this interaction the transcription activation domain will be brought in close proximity to the DNA binding domain, and as a result transcription activation takes place, and you can actually use reporter genes like beta galactosidase, and you can actually demonstrate blue color indicating that you have actually identified a particular protein which is actually interacting with this protein. So, this yeast-2 hybrid system became a very popular assay to study protein-protein interactions in eukaryotic cells. Okay. So, one of the major outcomes of demonstrating that the eukaryotic transcription factor possesses a modular structure with the DNA binding domain and transcription activation domain, they can be loosely tethered to each other, and that is sufficient for transcription activation is to understand and identify protein-protein interactions in eukaryotic cells. So, this is one of the important benefits that actually came out of all these studies. Now, so as people started studying more and more about the transcription activation domain function in eukaryotic cells, it became very clear there is nothing like a universal mechanism. Depending upon what kind of transcription factor you are studying and what kind of cell type you are studying, the regulation may be different. For example, there could be the function of a transcription activation domain could be modulated by binding of a small effector molecule. As I already told, in the case of proteins like glucocorticoid receptor, the binding of a steroid hormone actually modulates the activity of the glucocorticoid receptor. Now, the steroid molecule brings about a conformational change, dissociates the HSP90 in the cytosol, results in nuclear translocation and activation of transcription. There are actual instances where the transcription factor or the transcription activation domain function is actually modulated by specific post-translational modifications. It could be phosphorylation, acetylation, sumoylation, ubiquitination and so on and so forth. There are a number of post-translation modifications, and when these post-translation modifications happen, the transactivation domain function either can be activated or inhibited. We will discuss some of the examples later in this course. And there are also very important regulations where dimerization itself may play a very important role in the activity of the transcription factor. And this dimerization can happen between two same monomers when we call the results in the formation of homodimers, or it can actually form in the form of a heterodimer. Example can be the glucocorticoid receptor, for example, functions as a homodimer. So, two monomers actually come together, and through the dimerization interfaces they interact, and the dimer is actually goes and binds to the glucocorticoid response element and activate transcription. But in case of transcription factors known as C-Jun, C-Phos, or even last time we told NF-kappa B, which actually contains two different proteins which we discussed in the last class, there are actually the two monomers are not same, they are different. So, the dimerization can happen between two monomers of the same type or two different monomers, resulting in the formation of either homodimers or heterodimers, and that can also result in the activation of the transcription activation domain. So, there are a number of mechanisms by which the function of a transcription activation domain can be modulated. Another interesting aspect that actually emerged when people started making all these chimeric transcription factors and making recombinant transcription activation domains and starts looking at its function, especially in cell-free transcription systems, a very important observation that came out of these studies is that when cell-free transcription studies were carried out with purified RNA polymerase II and general transcription factors, it became very clear certain transcription factors or certain transcription activation domains require additional proteins. That is, in addition to the RNA polymerase II and general transcription factor, certain other proteins are actually required for the efficient transcription activation by certain eukaryotic transcription factors. For example, when people started purifying RNA polymerase II and people also started purifying the general transcription factors, and people in fact have cloned proteins like Tata binding protein, TF2B and so on and so forth, a very interesting experiment was done. People started doing what is called as a reconstituted in vitro transcription systems. We studied this in the couple of classes ago, that you can either use crude nuclear extracts or you can actually use reconstituted systems where the components are either purified from to almost 80 to 90% purity from various cell extracts, or you can even use recombinant general transcription factors and then assay promoter activity in vitro. When they did these kinds of studies, it became very clear when you want to do basal transcription, when you want to assay basal transcription, you can actually substitute TF2D with TBP. Now, as I told you, TF2D is nothing but TBP and TATA associated TBP associated factors or TAFs, right. Now, if you want to just do assay the basal activity of a promoter, if you just have a recombinant TBP instead of TF2D, you can demonstrate basal activation. But in such an assay, if you want to put a transcriptional activator and then ask whether the transcription activator can activate transcription, the TBP could not sustain or could not promote activator dependent transcription, which means very clearly that the TF2D, which actually contains TBP and TAFs, the TAFs have a very important role in facilitating activator dependent transcription in cell-free transcription assay systems. So, it became very clear that TAFs, which are the key components of TF2D in addition to TBP, may actually function as co-activators. So, this is the first hint that actually came, that one of the major mechanisms by which these general transcription factors facilitate is that many components of the general transcription factors actually interact with certain eukaryotic activators, and this interaction actually promotes in rapid recruitment of RNA polymerase to the promoter, resulting in the enhancement of transcription. And it also became very clear that different transcription activators may require different co-activators. For example, the TF2D itself consists of a number of TAFs, and in some cases a particular TAF may serve as a co-activator, but in other transcription activators a different TAF subunit may serve as a transcription activator. So, the outcome of what I have told you so far is that to demonstrate activator dependent transcription in cell-free transcription assays, it became essential that TBP is not sufficient, you require TF2D. I am clearly telling that for certain transcriptional activators, TAFs, which are components of TF2D, actually function as co-activators, and they are actually required to facilitate interaction between the RNA polymerase II and the activator. So, eukaryotic activators do not bind the RNA polymerase and therefore do not directly recruit RNA polymerase to the promoter. Activators through their transcription activation domains may indirectly recruit RNA polymerase to the promoters by recruiting certain factors, which I just now called as co-activators, and therefore these co-activators serve as physical bridge between activators and RNA polymerase. So, this is one important paradigm that emerged from the mechanistic studies on transcriptional activation domains of a number of eukaryotic factors. The other interesting experiment, especially from the group of Mark Tashney in the late 80s and early 90s emerged, is that I told you so just before in the last slide that different eukaryotic activators may actually require different co-activators, but there are also many examples where two different activators may require the same co-activator. Now, how do you demonstrate that? Some very interesting experiments were actually done, and these are actually called as activator interference or squelching experiments. Let me just explain to you what actually we mean by. Let us say, for example, there is an activator 1 and activator 2. These two activators bind to two different sequences. So, this is the DNA binding domain, transactivation domain for activator 1, DNA binding domain and transcription activation domain of transcription factor 2. Let us say, for example, this is GAL4 and this is GCN4. Okay, they bind two different sequences. Now, in a cell-free transcription system, if you now put purified GAL4, it will now go and bind to the GAL4 response element and activate transcription. Right, and let us assume that this transcription activation actually involves a specific co-activator, which I have shown in blue here. So, when the activator 1, in this case for example GAL4, binds to its sequence through the mediation of a specific co-activator, it now enhances the recruitment of RNA polymerase, resulting in the activation of transcription. But in such a transcription assay, if we now add excess amount of activator 2, now remember activator 2 cannot bind to the response element here, because the activator 2 cannot bind to the promoter here and activate transcription, because it has no binding domain. The DNA binding domain here is different. But if you now add excess amount of the activator 2, it actually, this activator 2 also requires the same co-activator which is required by activator 1, and therefore the co-activator being in limiting amounts got competed out by this excess co-activator, and as a result the co-activator was not available for transcription activation by activator 1. This actually resulted in inhibition of transcription. What actually it means that addition of excess co-activator here actually squelched the transcription activation by another transcription activator. Now, these kinds of experiments actually told you that two different co-activator proteins may actually be competing for the same co-activator proteins. So, there are instances where each transcription factor may require a different co-activator. There are also instances where both factors may be competing for the same co-activators, and these kinds of squelching experiments gave very new mechanistic insights into the co-activator requirements for very different transcription factors. For example, people actually, it became very clear there are a wide variety of co-activators. So, initially if you now look at the history of eukaryotic gene regulation, initially people started looking at general transcription factors, RNA polymerase and then core promoter elements. Then they realized that this alone is not sufficient, you require further upstream sequences either in the form of enhancers or distal promoter elements or proximal promoter elements. Then people started looking at protein factors which are actually binding to this. A wide variety of transcription activators started discovered. Then people looked at the mechanism of transcription activator function. People identified that they have different DNA binding domains, different transcription activation domains, and now people have realized in addition to all these things there are also a unique set of proteins called co-activators which actually serve as a bridge between transcriptional activators and the basal transcription machinery. In fact, one of the first co-activators which are actually identified in yeast cells is a protein called mediator, which is a multi-subunit complex. So, for many yeast transcription activators, this mediator served as a co-activator, and the mediator actually acted as a kind of a bridge between the activators and the general transcription factors for enhancing transcription activation. So, mediator is an essential component for RNA polymerase to general transcription machinery. It played a very crucial role in the activation and repression of many eukaryotic messenger RNAs in yeast cells, and the Saccharomyces cerevisiae mediator was the first to be identified, the first co-activator to be identified, and it has a very high molecular weight and is again a multi-subunit complex comprising of more than 20 distinct subunits. So, you see the complexity of eukaryotic gene activation. Already the basal transcription itself consists about more than 50 different polypeptides. Then I told you that you have other sequences which are play further important role in transcription activation, and there are a number of transcription factors with their own DNA binding domains and transactivation domains which are essential for transcription activation. And now I am telling you there are another unique set of proteins called co-activators which actually act as a bridge between eukaryotic activators and the basal transcription machinery, and they again are diverse and many of them are actually complex proteins with a number of protein components. So, over the years, especially in the 1980s and 1990s and even early part of 2000, people started looking at the DNA binding domains of various transcription factors. People also started looking at the transcription activation domain of various transcription factors, and it became very clear that each transcription factor is unique. Okay. Although there are some common motifs have been identified, like I told you, there some of them contain acidic domains, some of them contain glutamine domains, and so on and so forth. Many times it became very clear that as you start studying more and more transcription factors, it became more difficult to generalize. There are many of them which are very, very unique. Okay. So, what I will do is to actually just give you a very brief list of about 6 different transcription factors and briefly tell you what kind of transcription activation domains these proteins actually possess and how people went on studying these things. Let us take for example SP1. Now, SP1, as I told you already, contains a glutamine rich transcription activation domain, and a series of experiments actually demonstrated that the transcription activation domain of SP1 actually interacts with one of the TBP associated factors of TF2D called TAF2110. A 110 kilodalton TAF protein of the TFD complex actually facilitates the interaction between the SP1 and the general transcription machinery. So, TAF2110 is a co-activator for SP1. This was what I identified. If you come to steroid hormones, again the situation is very complex. Just to recapitulate, I told you already steroid hormones are lipid small molecules, they can actually diffuse into the cells. But in the absence of steroid hormone, the glucocorticoid receptor is actually present in complex with heat shock protein 90, which I have shown here. The white component is the GR and the black component is the HSP90 here. And once the steroid hormone diffuses into the cell and binds to the glucocorticoid receptor, it promotes dimerization of the 2 monomers of the GR and also dissociate the HSP90 from the glucocorticoid receptor, and as a result of this the nuclear localization signal on the present in the ligand binding domain is exposed, and therefore the receptor now translocates in the nucleus, binds to what are called as glucocorticoid response elements in the promoters and activates the transcription of the various genes. Okay. So, this is another example where a ligand actually modulates the activity of the transcription factor. Very interestingly, in the case of glucocorticoid receptor, in addition to the DNA binding domain and transcription activation domain, a very important domain called a dimerization domain was actually identified, and in fact this was found to be localized in the second zinc finger of the DNA binding domain. Remember, in the previous class I told you when you were studying the DNA binding domains, the zinc finger in the glucocorticoid receptor is of C2C2 type. It contains C2 type of a zinc finger, and it contains 2 such zinc fingers, and which are actually involved in the DNA binding. And I also told you the amino acids in the base of the first zinc finger play a very important role in DNA recognition, and by actually mutating these amino acids you can actually change the DNA binding specificity of glucocorticoid receptor. But now a specific amino acid, especially the amino acid 458 present somewhere at the base of the second zinc finger of the glucocorticoid receptor, if you for example mutate this amino acid, which is actually alanine, if you now mutate it to threonine, the receptor can no longer dimerize. So by simply making 1 point mutation in the dimerization in the second zinc finger of glucocorticoid receptor, you can generate a dimerization defective glucocorticoid receptor. So in addition to DNA binding domain, transcription activation domain, the DNA binding domain of GR also contains a very important dimerization domain, and there are also cases where dimerization domains are actually present in the transcription activation domains of other transcription factors. I will now give one example where the transcription activation domain function can be modulated by phosphorylation. Now there are proteins called as STATs. These are called as signal transducers and activators of transcription. Again, I am just giving a very brief overview here. As the course progresses, at a later stage we are going to take each one of these examples and study in detail how actually all these transcription factors function. Right now I am giving you only a very brief overview just to give you an idea of the diversity of regulation that is happening in the case of eukaryotic transcription factors, and the variety by which the function of transactivation domains can be regulated. Now many hormones, see in the previous case the glucocorticoid hormone, it actually diffuses into the cell and binds to an intracellular receptor, but there are many other hormones which actually do not enter. They actually, for example, growth factors. There are many growth factors which are polypeptides. They cannot just like that enter into the cell. They actually interact with specific cell surface receptor. For example, if you take epidermal growth factor, it actually goes and binds to a surface receptor called as epidermal growth factor receptor. And when these hormones, these polypeptide hormones bind to this cell surface receptor, it results in what is called as initiation of a signal transduction cascade. Now what do you mean by signal transduction cascade? A signal transduction pathway or a signal transduction pathway is nothing but when a specific ligand binds to an extracellular domain of a specific cell surface receptor, the binding brings about an allosteric change. Just like the glucocorticoid receptor binding to the glucocorticoid receptor in the cytoplasm brought out an allosteric change or a conformational change in the receptor so that it can no longer bind HSP90 and therefore it can dimerize and go into the nucleus. In the same manner, when certain polypeptide hormones bind to cell surface receptors, this ligand receptor interaction induces a conformational change in the cell surface receptor, and this conformational change ultimately results in the activation of a series of protein kinases and ultimately results in the phosphorylation of specific transcription factors, and they then go and bind to specific response elements and activate transcription. Okay. So phosphorylation is one of the key mechanisms in many cases in many signal transduction pathways by which specific transcription factors can be activated. I will give you one example. A very important cytokine which is involved in immune response is interferon gamma. Now when interferon gamma is added to the cells, okay, the interferon gamma actually goes and binds to a specific cell surface receptor, and this binding to the cell surface receptor triggers the phosphorylation of a protein called STAT1. Now the STAT1 is actually in an unphosphorylated form is present as a monomer and is actually present in the cytoplasm. Okay. Now once the interferon goes and binds to a cell surface receptor, it activates a protein kinase called Janus activated kinase or JAK kinase, and this JAK kinase now goes and phosphorylates the STAT protein, and once the STAT protein is phosphorylated, especially in the tyrosine residues, so the JAK kinase is a tyrosine kinase, so it phosphorylates specific tyrosine residues of the STAT1 protein, and this results in the formation of a homodimer. So phosphorylation of the STAT1 protein results in the homodimerization of the STAT1. Now this homodimer goes into the nucleus, binds to specific response elements called as interferon response elements, and activates a various genes which actually manifest interferon gamma response. Okay. So here the function of this transcription factor is modulated by phosphorylation, and this is actually because of a signal transduction cascade that initiated the cell surface and ultimately culminated in the phosphorylation of a transcription factor. So there are two signal transduction pathways. In cases like glucocorticoid receptor, the signaling molecules actually enter the cell and then modulate the function of a intracellular transcription factor. There is another signal transduction pathway where the molecules stay outside the cell, interact with specific cell surface receptor, and this interaction results in a initiation of a series of phosphorylation cascade, ultimately culminating in the activation of specific transcription factor inside the nucleus. There is another very interesting example. For example, in the case of HIV, it has a protein called as TAT protein. Now, HIV, as you know, is a retrovirus, and it is a very complicated retrovirus. Normally retroviruses contain only 3 proteins called GAG, POL and envelope, but HIV is a complex retrovirus. There are a number of other accessory proteins, okay, in addition to these 3 proteins, and one of the very important proteins that HIV contains, which is relevant to the gene regulation topic which is being discussed, is a protein called TAT. Now, this TAT is actually required for productive HIV gene expression. If the HIV genome has to be expressed efficiently, you require the TAT protein. Now, let us see what is the function of TAT protein. It turns out the TAT protein actually binds to an RNA stem loop structure called TAR, which is actually present in the 5' UTR of all HIV RNAs after the HIV transcription start site to regulate the level of transcription elongation. So far we have been discussing only about regulation of transcription initiation. Here there is a transcription factor which actually binds to an RNA and affects the regulation is at the level of transcription elongation. It turns out when there is no TAT, the HIV transcripts terminate prematurely due to poor plasticity of RNA polymerase II complex. So the transcription is initiated, but it does not proceed further, therefore it gets stuck. So the HIV RNA will not get transcribed efficiently because elongation does not take place, initiation takes place and the RNA polymerase does not move further. Now, in such a case, when TAT is expressed, the TAT actually binds to the star region in the 5' region of this HIV RNA, and because of this it recruits certain cellular RNA binding proteins to the nascent transcript DNA complex, and as a result of this the kinase activity of the TF2H is activated, and this results in the phosphorylation of the C-terminal domain of the RNA polymerase II, which, if you remember in my first class, phosphorylation of CTD is actually the signal for transcription elongation. It is the signal by which it is the RNA polymerase can now leave the initiation site and proceed further. So here is a viral transcription activator protein which actually binds the specific sequence in the viral RNA and then recruits a number of host transcription factors, ultimately resulting in the activation of the RNA polymerase II phosphorylation, so that the initiation complex can now leave the promoter and the RNA, the viral RNA can now be transcribed. So here you have a regulation having at the stage of transcription elongation. Another very interesting example I can tell you is a protein called MyoD, and if you remember from my previous class, MyoD actually contains a very interesting DNA binding domain called helix loop helix protein. Okay, the helix loop helix actually is again a very important dimerization motif where it actually brings the 2 subunits together so that it can actually go and bind to the DNA of a specific DNA sequence. Now, the MyoD was first identified as a gene that regulates the expression of cell determination and required for muscle differentiation. If you want the fibroblast to differentiate into skeletal muscle, you actually require this particular transcription factor. In fact, MyoD protein has been shown to activate muscle specific gene expression program. So if a particular cell type has to differentiate into skeletal muscle, the MyoD transcription factor plays a very important role. So if you now overexpress MyoD, it can actually turn fibroblasts into muscle like cells which express now specific muscle gene markers. So MyoD can actually change the differentiation program of a fibroblast into that of a muscle cell. That is because MyoD can go and bind to various genes which are actually involved in the muscle differentiation. In addition to MyoD, there are 3 other proteins called myogenin, MIF5, MRF4. They have all been shown to have the ability to convert fibroblasts into muscle. So these are all transcription factors which play a very important role in muscle differentiation program. Okay, and they all belong to this family of helix loop helix transcription factor family. Now, very importantly, there is a protein called ID which actually lacks a DNA binding domain, but it contains only the HLS dimerization domain, and when this ID protein binds to MyoD or related proteins, the heterodimers cannot bind DNA and therefore cannot regulate transcription. So this is actually called as dominant negative kind of a regulation. That means here you have a protein which has only the dimerization domain but does not have the DNA binding domain. So the heterodimer that is formed actually is defective and therefore it cannot act as a transcription factor. other very important example is what is called as during embryonic development, there are a bunch of transcription factors which are called as homeodomain protein. Again I discussed in the previous class that is homeodomain proteins actually contain a motif related to the helix turn helix motif which are actually present in the prokaryotic transcription factors, right. So these homeo box which actually contain a helix turn helix motif, DNA binding motif, which in this case they are often referred to as the homeodomain. So these transcription factors which contain homeodomain play a very important role during development and differentiation. For example, in the case of Drosophila, a specific homeotic gene called antennapedia, if you now mutate or if you now delete this particular region, you now mutate this, usually you have antenna in the head of the Drosophila fly, and instead of antenna you actually get legs. So this is what is called homeosis. There are actually transcription factor which can change the developmental program or differentiation program such that instead of formation of an antenna, a leg is formed in the head. So the whole organ transformation is actually controlled by a single transcription factor. We will study a little bit more about some of these things in the later classes when we start discussing about developmental regulation of gene expression during embryonic development. The very interesting aspects have come out of it. Just to mention here that there are transcription factors like homeodomain containing proteins or homeobox genes which actually can transform one organ into another organ if you have mutations in these transcription factors. So I think what I told you so far in this particular class, I just gave you a brief idea about what kind of transcription activation domains are actually present in various eukaryotic transcription factors. Okay, I told you there are many transcription factors which contain acidic activation domains, some of them contain proline rich domains, some of them contain glutamine rich domains, and a very key observation that came out of all these studies is that the transcription activation domain function can be separated from the DNA binding domain function. In fact, there have been a number of experiments that have been carried out wherein the transcription activation domains of one transcription factors were linked to the DNA binding domain of another transcription factor, and such chimeric transcription factors have been used to understand the mechanism of both DNA binding as well as transcription activation functions. And I also told you one of the important outcomes of all these experiments have been to actually develop what is called as a yeast 2 hybrid assay, wherein a protein is used as a bait and it is linked to the DNA binding domain of one particular transcription factor, and proteins which are likely to interact with this particular protein is linked to the transcription activation domain of other transcription factors, and we now intend to co-express these two proteins together. The transcription activation domain is brought in close proximity to the DNA binding domain, or this tethering of these two domains results in the activation of a reporter gene, and therefore you can actually conclude that this is the protein that is actually interacting with your protein of your interest. So the yeast 2 hybrid system was a very important outcome of understanding the modular nature of DNA binding domain and transcription activation domain of various eukaryotic transcription factors. And I also told you in these examples, I gave you various examples like steroid hormone receptors, homeobox proteins, and the yeast GCN4 and GAL4 and so on and so forth, just to give you an idea about the variety in the mechanism by which transcription activation domains function. And the other important point I told you in this class is that in order for these transcription activators or the transactivation domains to function or to activate transcription, in many cases you require what are called as co-activators. So these co-activators actually mediate or act as mediators between the transcriptional activators and the general transcription machinery. Now what we will do in the next class is to study a little bit more about the discovery of co-activators. Okay, and as we proceed further in the future classes, so far I have been discussing only about DNA. Now I have been talking as if the gene expression is all taking place on a naked DNA. Remember, in eukaryotic cells DNA is packed in the form of a chromatin. So in fact the most interesting aspect of this lecture series we will enter when we start looking at gene regulation that takes place in chromatin templates. We are now going to bring in at the stage histones into the picture. So far we have not talked about histones at all. So far we talked about core promoter elements, we talked about general transcription factors, then we talked about transcription factors and upstream activation sequences, then we discussed about the DNA binding domain and transcription activation domain of the eukaryotic transcription factors, and in this class I also brought you the concept of co-activators which act as mediators between the transcription activators and general transcription factors. But so far all this discussion centered around as if the DNA is a naked template, but which is not the case. In eukaryotes DNA is compacted in the form of a chromatin and histones are actually studied all over the DNA. So in the maybe about 2 or 3 classes later we will now start discussing how actually all these promoter elements, namely the core promoter elements, enhancer elements, the activator proteins, co-activators, all these functions have to be now take place on a chromatin template. and that is when we are now going to bring histones, and we are going to study what is called the histone code hypothesis, how modification of histones actually play a very important role in the regulation of gene expression. So, I think we still have not entered the most interesting aspect of this lecture series, and as we now start studying chromatin templates, we will actually understand a very important concept, maybe how all these co-activators and transcription activators, the mechanism by which all these things actually activate transcription is actually by modulating the function of histones. The transcription activators actually facilitate loose binding of histones, the transcription repressor facilitate tight binding of the histones, and it is by modifying the histone DNA binding activity that entire gene regulation takes place in eukaryotes. I think we will discuss some of this aspect as we proceed further in a couple of more classes, but in the next class we are going to discuss a little bit about co-activators and co-repressors and how these were identified and what kind of examples are actually present till today in the case of eukaryotic gene regulation. I think I will stop here.